सो हेलो एवरीवन, पिछले लेक्चर में हम लोगों ने पढ़ा था, समझा था कार्बोहाइड्रेट्स के बारे में, जैसे कि मोनोसैकेराइड है, ओलिगोसैकेराइड है, उनके बारे में डिटेल में समझा था। आज के लेक्चर में हम लोग पढ़ने वाले हैं, जैसा आप देख पा रहे हैं, पॉलीसैकेराइड यानी कि ग्लाइकन्स के बारे में। सबसे पहले सैकेराइड का मतलब समझ लेते हैं। सैकेराइड का मतलब होता है शुगर, और पॉलीसैकेराइड अगर वर्ड यूज किया गया, इसका मतलब कई छोटे छोटे जो मोनोमर है शुगर के, वो मिलके इस स्ट्रक्चर को, इस कंपाउंड को, इस पॉलीमर को बना रहे हैं, जो कि पॉलीसैकेराइड है या ग्लाइकन है। ठीक है, तो हम लोग थोड़ा स्टार्ट करने से पॉलीसैकेराइड को, उससे पहले थोड़ा रिवीजन करेंगे ओलिगोसैकेराइड के बारे में। जैसे कि हम लोगों ने देखा था कि टाइप्स ऑफ ओलिगोसैकेराइड, यहाँ पे हम लोगों ने डाइसैकेराइड के बारे में डिटेल में स्टडी किया था, क्योंकि हमारे इस नीट के लेवल पे डाइसैकेराइड पे ज्यादा जोर दिया गया है। कई जगह इसका यूज है, जैसे कि बायोमोलिक्यूल्स में इसके बारे में डिस्कस किया गया है, डाइजेशन चैप्टर में इसके बारे में डिस्कस किया गया। यहाँ कुछ डाइसैकेराइड हैं जो आपको यहाँ दिख रहे हैं स्क्रीन पे। पहला है सुक्रोज, जो कि किससे बना हुआ है? अल्फा ग्लूकोज और बीटा फ्रक्टोज। सिंस यहां अल्फा ग्लूकोज में फर्स्ट जो कार्बन रहता है और बीटा फ्रक्टोज का जो फोर्थ कार्बन रहता है, वो बॉन्डिंग में, बीटा फ्रक्टोज का जो सेकेंड नंबर कार्बन रहता है, वो बॉन्डिंग में इन्वॉल्व रहता है। इसका फर्स्ट, इसका सेकेंड कार्बन बॉन्डिंग में आ गया है, इसीलिए ये जो शुगर है, सुक्रोज जो है, नॉन रिड्यूसिंग शुगर होता है। कैसा शुगर होता है? नॉन रिड्यूसिंग शुगर। अब आप सोच रहे होंगे कि नॉन रिड्यूसिंग शुगर से मुझे क्या लेना देना। एक्चुअली सुक्रोज नॉन रिड्यूसिंग है, इसका मतलब वो नॉन रिएक्टिव इन नेचर है, और वो ट्रांसपोर्ट हो सकता है प्लांट में। तो प्लांट में जो शुगर ट्रांसपोर्ट होता है, वो किस फॉर्म में होता है? सुक्रोज के फॉर्म में होता है। स्टोर होता है स्टार्च के फॉर्म में, लेकिन ट्रांसपोर्ट होता है सुक्रोज के फॉर्म में। फिर दूसरा आप देख रहे हो माल्टोज। माल्टोज किसका डाइसैकेराइड है? 2 अल्फा ग्लूकोज का, और वो बॉन्डिंग जो होती है 1 और 4 नंबर कार्बन, एक इस ग्लूकोज के 1 नंबर, इसके 4 नंबर से क्या होगी? बॉन्डिंग होगी। और यहां जो बॉन्ड बनता है 2 ग्लूकोज को के बीच में, वैसे बॉन्ड को क्या कहते हैं? ग्लाइकोसिडिक लिंकेज। वैसे लिंकेज को क्या कहते हैं हम लोग? ग्लाइकोसिडिक लिंकेज। यहां आप देख पा रहे हो ट्रेहलोज। ट्रेहलोज किससे बना है? अल्फा ग्लूकोज और अल्फा ग्लूकोज से। ठीक है, फिर सेलोबायोज, ये सेलुलोज को डाइजेशन करने के बाद हमें सेलोबायोज मिलता है। आप देख पा रहे हो जो सेलुलोज पॉलीमर होता है, वो बीटा ग्लूकोज का होता है। ठीक है, फिर उसके बाद लैक्टोज, जो कि मिल्क शुगर है, बीटा गैलेक्टोज और बीटा ग्लूकोज के कॉम्बिनेशन से हमें मिलता है ये शुगर, जो कि मिल्क शुगर है, लैक्टोज। और आपको याद होगा जो मैंने ब्रेन शुगर की बात की थी, नर्व शुगर की बात की थी, वो क्या है? गैलेक्टोज। और आपको ये चीजें यहां तक क्लियर हो गई होगी। ठीक है, आगे हम लोग पढ़ेंगे इसके बारे में। किसके बारे में? पॉलीसैकेराइड के बारे में। चलिए अब स्टार्ट करेंगे। हमारा रिवीजन हो गया, ओलिगोसैकेराइड समझ लिया हमने, यहां हम पॉलीसैकेराइड के बारे में जाने। सबसे मोस्ट कॉमनली एनकाउंटर्ड कार्बोहाइड्रेट इन नेचर। आपको कार्बोहाइड्रेट ग्लूकोज के फॉर्म में हमेशा नहीं मिलेगी, कार्बोहाइड्रेट हमेशा सुक्रोज के फॉर्म में नहीं मिलेगी। सुक्रोज के फॉर्म में आपको मिल सकते हैं, चीनी वगैरह मिल सकते हैं, ठीक है, लेकिन हमेशा नॉर्मल जो ज्यादातर आपको उस फॉर्म में नहीं मिलेगा। आपको किस फॉर्म में मिलेगा? ये पॉलीसैकेराइड या ग्लाइकन के रूप में आपको कार्बोहाइड्रेट ज्यादा मिलेगा। ठीक है, ध्यान देना, ये क्या है? इट कंटेन्स अ लार्ज नंबर ऑफ मोनोसैकेराइड। ये क्या कंटेन करता है? लार्ज नंबर है मोनोसैकेराइड का यूनिट्स, और विच इज ज्वाइंट टुगेदर। सारे मोनोसैकेराइड, जैसे डाइसैकेराइड में 2 मोनोसैकेराइड जुड़ते थे ग्लाइकोसिडिक लिंकेज से, वैसे यहां कई मोनोसैकेराइड जो है आपस में जुड़ेंगे ग्लाइकोसिडिक लिंकेज से। किस तरह के लिंकेज से? ग्लाइकोसिडिक लिंकेज। इस चीज को आप समझ पा रहे हो? ठीक है, अब लास्ट में देखिए, राइट एंड इज कॉल्ड द रिड्यूसिंग एंड। जो ये आपका पॉलीसेक्राइड है, इसका जो राइट एंड होता है उसे क्या कहते हैं? रिड्यूसिंग एंड। एंड द लेफ्ट एंड इज कॉल्ड नॉन रिड्यूसिंग एंड। इसका लेफ्ट एंड जो होता है वो क्या कहलाता है? नॉन रिड्यूसिंग एंड। राइट एंड क्या है? आर से राइट एंड आर से रिड्यूसिंग एंड, ठीक है। और दूसरा है नॉन रिड्यूसिंग एंड, और दूसरा जो एंड होता है, लेफ्ट का एंड होता है, उसे नॉन रिड्यूसिंग एंड कहते हैं। हेलो फैन क्लब, सॉरी फन क्लब, क्या नाम है आपका? चलिए यहां पे समझते हैं ये पॉलीसैकेराइड कितने तरह के हैं। यहां क्लासिफाई किया है हमने पॉलीसैकेराइड को। सबसे पहले यहां आप देख रहे हैं होमोग्लाइकन और होमोपॉलीसैकेराइड, फिर उसके बाद यहां आप देख रहे हैं हेट्रोग्लाइकन और हेट्रोपॉलीसैकेराइड। इस चीज को समझते हैं। ये होमो का वर्ड, होमो का मतलब मैं समझा देता हूं यहां पे, कि एक तरह के मोनोमर से बना हुआ रहता है। एक तरह के, जैसे मान लेते हैं एक तरह का मोनोमर होता है, ग्लूकोज ले लेते हैं, तो कई सारे हजारों 2000 ग्लूकोज को आप जोड़ते जाओ ग्लाइकोसिडिक बॉन्ड से, तो जो स्ट्रक्चर बनेगा वो होमोपॉलीसैकेराइड बनेगा। और ये हेट्रोपॉलीसैकेराइड या हेट्रोग्लाइकन क्या होता है, इसके बारे में हम लोग बात करेंगे, ठीक है। हियर वी हैव समर। चलिए समर पढ़िए। अगर आपको कहीं भी कुछ डाउट आता है तो आप पूछ सकते हैं यहाँ कमेंट सेक्शन में, या फिर किसी चीज को आप समझ नहीं पाए हैं, उसे अगर रिपीट करना है तो वो भी आप बता सकते हैं मुझे यहाँ कमेंट सेक्शन में, ठीक है। तो हम यहाँ पे होमोग्लाइकन और हेट्रोग्लाइकन के बारे में समझ रहे हैं, ठीक है। अब यहाँ पे होमोपॉलीसैकेराइड के बारे में, यानी कि होमोग्लाइकन के बारे में बता रहा हूं। अब यहां फन क्लब कर रहे हैं, हाउ कैन यू टेक ऑल दिस क्लासेस विदाउट प्रॉपर व्यूअर्स। व्यूअर्स अपने आप जुड़ जाएंगे जिन्हें पढ़ना होगा, है कि नहीं। अब यहां पे होमोपॉलीसैकेराइड, जिस तरह आप जुड़े उस तरह कोई और भी जुड़ जाएगा, जिनको सीखना होगा, जितना पढ़ना होगा, जिन्हें पढ़ना होगा, है कि नहीं। और ये एक तरह का ना एक ट्रायल क्लास चल रहा है कि टू सी द रिस्पॉन्स ऑफ द व्यूअर, ठीक है। यहां देखते हैं होमोपॉलीसैकेराइड। सबसे पहले हम लोग देख रहे हैं होमोपॉलीसैकेराइड फॉर्म्ड बाय पॉलीमराइजेशन ऑफ वन टाइप ऑफ मोनोसैकेराइड यूनिट। तो वी हैव राहुल कुमार आल्सो हियर। ये जो आपका होमोपॉलीसैकेराइड है वो कैसे बनता है? एक तरह के वन टाइप ऑफ मोनोसैकेराइड यूनिट को आप जब जोड़ देते हो तो आपका होमोपॉलीसैकेराइड बनता है। डिपेंडिंग अपॉन द मोनोसैकेराइड यूनिट, किस तरह के मोनोसैकेराइड यूनिट को आपने जोड़ा है, ठीक है, उससे डिपेंड करता है उस पॉलीसैकेराइड का नाम क्या है, ठीक है। हाय राहुल, देखिए समझने की कोशिश कीजिए, अगर कुछ डाउट है तो जरूर पूछिए। यहां आप देख पा रहे हो कितने तरह के पॉलीसैकेराइड है? ग्लूकन, फ्रक्टन, गैलेक्टन, एराबन। Fun Club is telling me it will be good if you take class in the English. If you want me to take class in English, I will deliver the class in English. See here. Here we have the... Okay, you are from South India and very weak in India. Fine, I will take class in English today. So here we have different polysaccharide like glucan, fructan, galactan and araban. Glucan is a polysaccharide which is made up of monosaccharide known as glucose. Fructan is a polysaccharide which is made up of fructose, and the galactan is the polysaccharide which is made up of galactose. And here we have araban. It is also polysaccharide which is made up of arabinose. So here these all are monosaccharide which keeps on repeating itself in the chain to form these types of polysaccharide respectively. Okay. राहुल कह रहे हैं कि सर अभी तो डाउट नहीं है। अच्छी बात है, कोई डाउट नहीं है। ओके, here we have heteropolysaccharide. Now we will understand what is the meaning of heteropolysaccharide. We understood the meaning of homopolysaccharide. See here, produced by, what is produced by? Heteropolysaccharide is produced by condensation of either monosaccharide derivative or more than one type of monosaccharide unit. Let me explain you or explain you the difference between heteropolymer or heteropolysaccharide and homopolysaccharide. Homopolysaccharide was made up of only one type of monosaccharide, but here it is made up of more than one type of monosaccharide. Like it can, if you see the polysaccharide that is heteropolysaccharide, when you break it, you will get glucose also, you will get fructose also, or it is made up of the derivative of monosaccharide, and we studied about the derivative of monosaccharide in the previous lectures, and you can see there. Okay, see here we have the polysaccharides. It is divided. First we divided the polysaccharide or this glucose or the carbohydrate, the complex carbohydrate, into homopolymer and heteropolymer. We have the another classification of polysaccharide or glycans, and here these are food storage polysaccharide. That is the polysaccharide which is used as stored food. See here we are using the food storage, which is in the polysaccharide which is used as food, which is used as polysaccharide. Here we have a comment. Are you using green screen there? Yes, we have the green screen here. Are you facing problem while seeing the lecture? So here we have the one carbohydrate which is used as stored food. We have the structural polysaccharide, that is which provide structural, like chitin, and you know that chitin is the exoskeleton of which organism? All the arthropoda. Okay, we have the cellulose. Cellulose is the cell wall of all the plant cell, agar. Okay, and then we have the Muco Polysaccharide. Muco Polysaccharide is also a type of polysaccharide, and the examples are hyaluronic acid, heparin, chondroitin sulfate, keratin sulfate. Okay, hyaluronic acid, we have the hyaluronic acid in our joints. Okay, and it is also known as cement of animal cell. Heparin, heparin is a mucopolysaccharide which does not allow our blood to clot easily in the normal condition, although we need the clotting of blood when there is a cut. Okay, but during the normal circulation we don't want our blood to clot, otherwise there will be problem. So there we have the heparin, and then we have the chondroitin sulfate. Chondroitin sulfate is present in the cartilage. Then we have the keratin sulfate here, which is present in our hair, our nails. Okay, so these are the polysaccharide, the types of polysaccharide which we are going to study later in the lecture. Fine. Now we will see the food storage polysaccharide one by one. First we will see the storage polysaccharide, that is starch. Please pay attention. Carbohydrate like glycogen and starch. There are two carbohydrates. Please see here. Glycogen and starch. Very important. Glycogen and starch are easy to store because of the following advantage. See, glycogen and starch are non-reactive and non-soluble polysaccharide. So they are stored as a stored reserve food. Starch is stored as a reserve food in the plant, and glycogen is used as a reserve food in animals. Am I clear? And what are the features or what are the qualities which is allowing them to become the storage polysaccharide? Let me make you clear. Here we have the chemically non-reactive polysaccharide. Yes, the glycogen and starch are chemically non-reactive. And the second one, it is osmotically inactive. Osmotically inactive means it is not dissolvable, it is not soluble. Okay, it cannot dissolve in water, so that's why it can be stored as a stored food, and then it can be stored in bulk. Yes, these are the qualities which is allowing or which is making this glycogen and starch as a storage polysaccharide. Is there any doubt till here? Is there any doubt till here? Can anyone tell me what is carbohydrate? See here we have different storage polysaccharide. We studied the most important one, that is the starch and glycogen, but we have some more storage polysaccharide here. These are starch, glycogen, inulin, dextrin and dextran. We are going to study about them one by one. Although we studied, we know the introduction about these structures, these polysaccharides. First, we will deal about the starch, that is amylum. Its other name is amylum. And you know, the enzyme which help in the digestion of starch is known as amylase. Amylum, amylase. And the other name for amylase is ptyalin. Am I clear? Am I clear? Can you tell me which gland in our oral cavity secretes the enzyme amylase? Can you tell me which gland in our oral cavity secrete the enzyme amylase? The main storage starch here. Main storage polysaccharide of the plant. Yes, this starch is the main storage polysaccharide of plant. Most important dietary source for human being. Yes, this is the most important dietary source for human beings. High content of starch is found in cereals, tuber, root and some vegetables. See where these starch are present. These starch are found in cereals, tuber, roots and vegetables. Forms helical secondary structure. Yes, this has the helical secondary structure present, and that's why it gives the iodine taste. You must have seen the iodine taste of the starch, and this is the reason why this helical secondary structure holds the iodine, and that is the reason starch gives the positive test for iodine. It can hold the molecule in its helical portion. Starch iodine is blue in color, and starch is made up of two types of polysaccharide, that is amylose and amylopectin. We will know about the amylose and amylopectin in the further slide, but now we have to learn these two words, that is it is amylum or starch is made up of two types of polysaccharide, that is amylose and amylopectin. Fine. See, this is the structure of amylose. This is here we have the starch. Can you see this structure here? This is the right side of the starch and this is the left side of the starch. Am I clear? And this is made up of amylose and amylopectin. The structure which you can see here is amylose because it is unbranched. I will show you how the branching is there. Here we have the amylose and amylopectin difference. See, amylose is the polymer of 200 to 1000 alpha D glucose. Okay, amylose is a polymer of 200 to 1000 alpha D glucose, while amylopectin it is a polymer of 2000 to 2 lakh alpha D glucose. Please let me make it clear. This is also the part of starch. This is also the part of starch. Amylose 200 to 1000 alpha D glucose mix this polymer amylose, and 2000 to 2 lakh amylopectin mix this polymer, that is amylopectin. Sorry, 2000 to 2 lakh molecule of glucose. Which type of glucose? Alpha D glucose. Mix the polysaccharide amylopectin, and both these structures, both these polysaccharide form starch. Here we have the another difference, unbranched and spiral chain compound. This amylose is unbranched in nature. Please see here, this is unbranched in nature, and it is spiral chain compound. It is what type of chain is found here? Spiral chain compound. The branched globular structure here in amylopectin, you will see the branched structure. You will see the branching in case of amylopectin. Branching occurs at every 25 to 30 glucose. Let me make it clear. After every 25 to 30 glucose, there is a branching seen in case of the amylopectin. Am I clear here? 24 to 30 glucose units. Okay, and here we have the amylose. Glucose molecules are interlinked by, these glucose molecules are interlinked by alpha 1,4 glycosidic bond, and here the linear part we have alpha 1,4 glycosidic bond, and the branching is due to the alpha 1 and 6 glycosidic bond, which we will see in the chemistry lecture. It is a water soluble compound, and here we have water insoluble compound. Amylopectin is branched in nature, and that is the reason it is not soluble in water. So if the question comes, is the starch soluble in water? No, it is not soluble in water. Am I clear? Only the amylose part of starch is soluble in water. Let me tell you one thing, that whenever you put a potato in water, you see the color of water changes into white. Why? The amylose part of the potato, the amylose part of the starch of the potato gets dissolved in water and change the color of water to white, while the amylopectin part of the starch of potato does not dissolve in water. So that's why you can see the amylopectin even after boiling. Okay, and the other difference, it constitutes 15 to 20% of the starch, and this amylopectin constitutes 80 to 85% of the starch. It gives blue color to iodine, this amylose, and it gives red color to iodine. If you separate the amylose and amylopectin, you will get these separate color test when you treat them with iodine. Here you have the blue color and here you have the red color. Is there any doubt till here? क्या यहाँ तक कोई doubt है? So we have another structure. यहाँ हमारे पास दूसरा स्ट्रक्चर है ग्लाइकोजन का. ठीक है, ग्लाइकोजन भी एक पॉलीसैकेराइड. इट इज ऑल्सो अ पॉलीसैकेराइड, एंड इट इज द पॉलीसैकेराइड विच इज फाउंड इन एनिमल. It is the storage polysaccharide which is found in animal. It is present in animal and also known as animal starch. Yes, this is known as animal starch. It is branch chain compound and has branching after 8 to 12 glucose. See, वहां सिर्फ amylopectin में branching था, but यहाँ पूरे glycogen में branching है. So it is branch chain compound and has branching after 8 to 12 glucose unit. It is stored mainly in liver. ये कहाँ stored रहता है? ये ऐसे पार्ट में stored रहता है जहां पे ग्रोथ हो रहा है, जहां मेटाबॉलिज्म हो रही है. ठीक है, तो लिवर में स्टोर हो सकता है. इट कैन बी स्टोर्ड इन लिवर, इट कैन बी स्टोर्ड इन मसल्स, इट कैन बी स्टोर्ड इन किडनी, प्लासेंटा. बट इफ only single option you have to select, the best option will be the liver. In large amount the glycogen is stored in liver. Am I clear? It is also the stored food of many fungi like yeast, and it gives red color with iodine. It also gives red color. You have seen the amylopectin of the starch is also giving the red, and glycogen is also giving the red color to the iodine. Am I clear? So we have these important lines. It is found in large amount in liver. So here we completed the starch and the glycogen. Can you see the branching? We don't have to learn all this, just see the branching, how the branching is there. Can you see the branching? Yes, this is the branching. Here we have the inulin. It is the polymer of fructose, and let me make it clear that this polymer of fructose, which is used in GFT or KFT, or sorry, not GFT, RFT, renal function test. Okay, it helps to see or check the glomerular filtration rate. It is a polymer of fructose. You cannot digest this inulin, so it is used to check or test the kidney. It is found in the roots of dahlia, artichoke, sweet potato, onion and garlic. It is water soluble polysaccharide. This polysaccharide is water soluble in nature. It is not metabolized in human body. You cannot digest them. Am I clear? You cannot digest the inulin and is readily filtered through kidney, so used in testing of kidney function test or KFT. You can also call it RFT, renal function test. We use this compound, that is inulin. Am I clear? Here we have the dextrine. Dextrine is partially digested starch. It is an intermediate substance in the digestion of starch and glycogen. Please pay attention. It is the intermediate substance in digestion of starch and glycogen. Glucose and maltose are formed by the hydrolysis of dextrine. Whenever there is an intake of food or starch, it is first converted into dextrine by the action of your salivary amylase. Salivary amylase, which is secreted from your salivary gland, works on which part? The starch, and then convert into dextrine, and then it again works on dextrine, and then dextrine is converted to maltose. Am I clear? And that maltose is sweet in taste. All the sugars, all the carbohydrate are not sweet in taste. These things like starch, glycogen are not sweet in taste, but maltose is sweet in taste. So glucose and maltose are formed by the hydrolysis of dextrine. It also occurs as stored food in yeast and bacteria. Please pay attention. But this is more important. Glucose and maltose are formed by the hydrolysis of dextrine. Am I very clear here? How the dextrine is formed? Is there any doubt till here? It also occurs as a stored food in yeast and bacteria. Let me ask some question. Glycogen is a polymer of amino acid. Glycogen is a polymer of, first option is amino acid, second option is glucose, third option is nucleic acid, and fourth is all of these. What should be the answer? Yes, it is a polymer of glucose. If someone asks you what is carbohydrate, so carbohydrate is a polymer of fatty acid. Okay, carbohydrate is a polymer of fatty acid, or carbohydrate is a polymer of amino acid, or carbohydrate is polyhydroxyaldehyde or ketone. What is carbohydrate? Is it polyhydroxyaldehyde or ketone? Yes, the carbohydrate is polyhydroxyaldehyde and ketone or ketone. Am I clear? We have another polysaccharide like dextran. It is the polymer of glucose produced by microorganisms. Am I clear? So they are used as plasma volume expanders in transfusion. This is not of our level, of NEET level. We will study about this later. We have some structural polysaccharide like the polysaccharide which is just giving the shape, the structure to any part, forming the structures like cellulose. You must have seen the cellulose is present in the cell wall of the plant cell. It is giving a structure to the plant cell. Chitin is also present in the cell wall of the fungus, and chitin is also present in the as an exoskeleton of insecta, arthropoda, and then we have agar. These are the structural polysaccharide which is giving a particular shape, particular structure to any part of the body or any cell. Am I clear? First we will know about cellulose, which is the most abundant carbohydrate in nature. Please note it down. It is one of the abundant organic compound in nature also. Cellulose is the most abundant organic compound in nature. It is the most abundant organic compound. Okay, let me repeat. See, it is a fibrous polysaccharide and forms cell wall in plant. First, very important, is a fibrous polysaccharide. Fibrous in nature. When you see it in microscope, you will see there is a fibers. Okay. Cellulose molecules have unbranched and linear chain of 6000 or more beta D glucose. We have studied about this beta D glucose. We know about alpha glucose and beta glucose. Here we have the polymer of beta glucose. Alpha glucose polymer is present in starch or glycogen, while beta D glucose polymer is cellulose, and mammals' own enzyme cannot digest beta D glucose polymer. Beta glycosidic linkage cannot be broken by any of the enzymes of the mammals. So, but you know that some of the ruminants, some of the cattle can graze on plants and can digest. In that, the digestion of that part of the plant is not done by the animal itself. The symbiotic bacteria which is present in the gut of those ruminant helps in the digestion of cellulose, and they have the ability to break the beta glycosidic bond of the cellulose into glucose, and that glucose which is digested by the symbiont inside the ruminant or the cattle forms the glucose, and that glucose is utilized by those cattle. So here cellulose molecules have unbranched and linear chain of 6000 or more beta D glucose, and it forms roughage in human food. So if someone asks you, do we need cellulose? Yes, we need cellulose. Although we can't get glucose from the cellulose, but still we need cellulose because it is the part of the roughage. And what is the function of roughage? आपका जो खाना पचता है, it helps in the digestion of the food. It allows the wall of the gut or digestive tract to secrete more mucus so that it prevents the constipation. And see, it forms 20 to 50% of the wool and about 90% of the cotton. Cotton is also fibrous in nature, and see here it is a fibrous polysaccharide. Cellulose is fibrous polysaccharide, and you can see here in cotton you have 90% of the cellulose. Cellulose is also present in the wood, 25 to 50%. In urochordates, you know about tunicates. There tunicin is present as a covering which is made up of cellulose. Tunicin is the covering of tunicates which is made up of cellulose. Cellulose do not contain complex helices like starch and hence cannot hold iodine, so it won't give blue-black color. Cellulose does not contain complex helices like starch and hence cannot hold the iodine. That's why cellulose will give negative test to iodine. Am I clear? So here we have completed the cellulose. Okay. Now we have the types of cellulose compound. First we have the cellulose acetate. Can you see here the cellulose acetate? Then we have carboxymethyl cellulose. Then we have the cellulose nitrate. These are the different compounds of cellulose and we will see their use. See, cellulose acetate is used in fabrics like cotton, cellulose plastics, shatterproof glass which don't breaks, then cigarette filters. This cellulose acetate is used. Carboxymethyl cellulose, it is used in ice cream, cosmetics, medicine to emulsify and give them smooth texture. Okay, and then we have the cellulose nitrate. It is used in propellant, explosive. Okay, these are also asked in the chemistry: cellulose nitrate, carboxymethyl cellulose, and cellulose acetate. Do you have any doubt in this part? If you have any doubt, you can write in the comments section. Here we have the another polysaccharide. It is the most abundant polysaccharide is or most abundant organic compound is cellulose, and this chitin is second most abundant organic compound. It is second most abundant organic compound. It is for polymer of N-acetyl glucosamine. We have studied about the N-acetyl glucosamine. It is modified or derived what? Derived monomer, derived monosaccharide. It is derived monosaccharide. It is a polymer of N-acetyl glucosamine. It is present in the cell wall of fungi. You know that chitin is present in the cell wall of fungi. Am I clear? Fungal cellulose, and there it forms the exoskeleton mainly in arthropods. Am I clear? It is a soft and leathery. What is the texture? It is soft and leathery, so provide strength and elasticity. It is soft and leathery, so it provides strength and elasticity. Do you have any doubt till here? Yeah. It hardens due to deposition of proteins and calcium carbonate. So you must have seen that there is a hardness in the exoskeleton of arthropoda. What is the reason? There is a deposition of proteins and calcium carbonate. वहाँ deposition होता है किस चीज का? प्रोटीन का और calcium carbonate का, and this is the reason chitin is hard in texture when you touch some of the touch some of the arthropods. Am I clear? So see here, these are the derived carbohydrate. Can you see this? On the second number carbon we have nitrogen containing compound. So this is, although the monomers are repeated, but still this is heteropolysaccharide because it is made by the repetition of the derived monosaccharide. Am I clear? And here we have the agar. यहाँ अब हमारे पास क्या है? agar है. ओके, इट इज अ नेचुरल कंपोनेंट ऑफ सीवीड्स. अगार जो है एक नेचुरल कंपोनेंट है सीवीड का. इट इज मेड अप ऑफ सल्फेटेड गैलेक्टोज यूनिट. ओके, दिस अगार इज made up of sulfated galactose unit. That sulfate is present in the galactose. In microbiology, it is used as a culture medium. Yes, what is culture medium? The place, the material, the medium in which you grow bacteria or other microbes in order to see their structure, see their nature, how they are growing, how they are working, effect of chemicals on them, effect of antibiotic on them. So this is the medium which provide the culture, the medium on which the microbes can grow. So agar is a medium on which microbes grow. Okay, it is obtained from two places, Gracilaria and Gelidium, or you must have studied about this Gracilaria and Gelidium in which chapter? In the chapter Plant Kingdom. So we have this agar is obtained from Gracilaria and Gelidium, and this Gracilaria and Gelidium are algae. Am I clear? So that's why it is the component of natural component which is found in sea. See here we have Muco Polysaccharide. What is Muco Polysaccharide? See, whenever you cut any lady finger, you get a sticky substance, and that sticky substance is Muco Polysaccharide. Not only in plants, even in our body, our whole digestive tract secrete a mucin, that is Muco Polysaccharide. Let me explain you what is this. Their building blocks are amino sugar. It is derived sugar. Amino group is attached to the sugar. Can you see here? Amino group is attached to the sugar here. Can you see this? Yes. This is a repeated, and this is a the monomer of mucopolysaccharide, and this structure is repeated in order to form mucopolysaccharide. So the building blocks are amino sugar and chemically modified sugar like glucosamine, N-acetyl glucosamine, galactosamine. These are commonly known as Glycosaminoglycans or GAG. These are also present in Vindi or Isabgol. You must have seen Isabgol. When you put it in the water, there is a sticky structure. There is a mucin-like structure is formed, that is mucopolysaccharide, and it is commonly known. It is made up of. This is commonly the chemical name of this mucopolysaccharide is glycosaminoglycan or GAG. Okay. Here we have the different kinds of mucopolysaccharide. These are hyaluronic acid, heparin, chondroitin sulfate, keratin sulfate, heparin sulfate, and dermatan sulfate. These are different acid. Can you see here? Hyaluronic acid, heparin, chondroitin sulfate, keratin sulfate, heparin sulfate, and dermatan sulfate. We will study about these in detail. First, let me make you clear that which are mucopolysaccharide which is important for our exam. That is hyaluronic acid, heparin, chondroitin sulfate, and keratin sulfate. These four are important for us because these four have been given in our NCERT at different places. Okay. फर्स्ट वी विल स्टडी अबाउट म्यूकोपॉलीसैकेराइड. हम लोग पढ़ने वाले हैं इस म्यूकोपॉलीसैकेराइड के एक फर्स्ट फॉर्म जो hyaluronic एसिड है. hyaluronic एसिड इज फाउंड इन द विट्रस ह्यूमर. ये कहां मिलेगा हमें? विट्रस ह्यूमर में मिलता है. देन इट इज ऑल्सो फाउंड इन अम्बिलिकल कॉर्ड. व्हाट इज अम्बिलिकल कॉर्ड? इट इज द कॉर्ड विच कनेक्ट्स placenta to the fetus. Umbilical cord. It is found in the joints. Yes, sometimes after the surgery of the joint, since there is a friction, so this hyaluronic acid is injected inside the joint to decrease the pain and to decrease the friction between the joints. And then it is also present in the connective tissue and is also known as binding material in animal cell or animal cement. See, this is important. This can come in NEET exam. What is the animal cement? What is the other name of the animal cement? Or which chemical, which mucopolysaccharide is animal cement? The answer will be hyaluronic acid. What is the answer? Hyaluronic acid. Am I clear? So this is important for NEET exam. Heparin is anticoagulant. We have the another mucopolysaccharide, heparin. As I explained earlier, what is heparin? Heparin is the anticoagulant which is present in our blood and prevents unnecessary clotting of blood in our body. Am I clear? And then we have the chondroitin sulfate which is present in cartilage, tendons, and bones. You must have studied this chondroitin sulfate in the chapter the structural organization of animals. Can you see the chondroitin sulfate? It is the component of the cartilage, tendons, and bones. Am I clear? Then we have the keratin sulfate which is found in costal cartilage and cornea. What is costal cartilage? Tell me, what is costal cartilage? Costal cartilage is present here. Here we have the sternum. Here we have the sternum and the ribs are attached to the sternum by the cartilage that is known as the costal cartilage, and the chemical which is present in costal cartilage or the compound which is present in the costal cartilage is keratin sulfate. And not only in costal cartilage, it is also present in the cornea. And what is cornea? What is cornea? The outer layer, the sclera. The outer layer of the eye is known as sclera. Then inside the sclera we have choroid. Inside the choroid we have retina. So the outer layer of sclera is transparent in the anterior region, and that is known as cornea. It is the non-vascular part and it is made up of keratin sulfate. Am I clear? And then we have the dermatan sulfate. Dermatan sulfate is present in the wall of the arteries. Dermatan sulfate कहाँ present होता है? हमारे artery के wall पे. Am I clear till here? So we have discussed everything about the mucopolysaccharide also. Okay. So let me explain some of the facts about the nucleic acid. We won't go in much detail of the nucleic acid because nucleic acid is a very big topic and it is a part of molecular basis of inheritance. There we are going to study them in detail. But we will have some introduction in this chapter, in this lecture. Nucleic acid are the biopolymer or large biomolecule essential to all known forms of the life. Yes, it can be virus, it can be, see, virus, although it's not the life, it's not the living thing, but still there you have the nucleic acid. And then in bacteria you have nucleic acid, in eukaryote, prokaryote, eukaryote, everywhere you have nucleic acid in the form of DNA or RNA. What is DNA? Deoxyribonucleic acid. And what is RNA? Ribonucleic acid. So this is nucleic acid. It can be deoxy or just ribonucleic acid. The nucleic acid is the overall name. Nucleic acid is the collective name for DNA and RNA. As I told you, what is DNA? Hello Ayush Mishra. So what is nucleic acid? Nucleic acid is a collective name for deoxyribonucleic acid as well as ribonucleic acid. They are composed of, when you see them, they are composed of nucleotides. What are they composed of? Nucleotides. That is, their monomers are known as nucleotides. Many nucleotides combine to form nucleic acid. कई सारे न्यूक्लियोटाइड्स मिलते हैं और क्या बनाते हैं? न्यूक्लिक एसिड. और न्यूक्लिक एसिड का कितना फॉर्म है? दो फॉर्म है, एक तो डीएनए है और दूसरा आरएनए है. आई बात समझ में? चलिए आगे समझते हैं पॉइंट्स. एंड दिस इज मेड अप ऑफ, ये जो न्यूक्लियोटाइड है ये तीन तरह के कंपाउंड से बना हुआ है, तीन मॉलिक्यूल से बना हुआ है. पहला देखते हैं 5 कार्बन शुगर, 5 कार्बन शुगर. दूसरा क्या है? फॉस्फेट ग्रुप. और तीसरा है नाइट्रोजनस बेस. हियर वी हैव three components. One is 5 carbon sugar, phosphate group and the nitrogenous base. If the sugar is composed of ribose, अगर इनका शुगर, यहां जो शुगर की बात हो रही है 5 कार्बन शुगर, अगर वो राइबोस का होगा तो उस पॉलीमर को क्या बोलेंगे? राइबोन्यूक्लिक एसिड. देन दैट पॉलीमर विल बी कॉल्ड राइबोन्यूक्लिक एसिड. बट इफ that is, we have discussed the ribose and deoxyribose. If in nucleotide we have deoxyribose in place of ribose, then the compound is known as DNA or deoxyribonucleic acid. With how care is chapter name is natural resources? No, this is the chapter name. The chapter name is biomolecule and it is of class 11. And we have discussed carbohydrate in detail till now. Now we are discussing the nucleic acid. See here, we are studying the component of nucleotide. Can you see here the nitrogenous base? This is the nitrogenous base. And can you see the sugar part? Yes, this is the sugar part. Can you see here there is no oxygen present on this carbon? So that's why this is deoxyribose, 5 carbon non-deoxyribose, 5 carbon compound. Okay, 5 carbon sugar, deoxyribose. And can you see the phosphate? And yes, when all these things, okay sir, he is telling, sorry sir, wrong class, I was going in 9th class. It's okay, fine. Here we have the nitrogenous base, then there is a sugar, and then here we have the phosphate. These three molecules combine to form the monomer of nucleic acid, that is a nucleotide. And many these types of nucleotides joins one to another and form whole nucleic acid like this. Please see here. Can you see the chain of nucleic acid? This is the chain of nucleic acid. This is the nitrogenous base. Can you see the sugar part? And this is the phosphate. Nitrogenous base sugar part phosphate, nitrogenous base sugar part phosphate, nitrogenous base sugar part phosphate. These are joined to one another and form a chain known as nucleic acid. And here we have the another chain of nucleic acid, and both these chain are joined by the bond known as hydrogen bond. So this is nucleic acid. We discussed about the nucleic acid. Am I clear? And can you see the types of nitrogenous base? Adenine, thymine, cytosine, guanine. These are the types of nitrogenous base. Please look here. Adenine, guanine, thymine, cytosine. These are the nitrogenous base which is present. Okay. Do you have any doubt till here? Can you see here this part, the animated part? Here you have the nitrogenous base. Nitrogenous base is joined to the sugar, and then sugar is joined to phosphate. And see how the different nucleotides are linked using phosphate. And we are going to discuss about this in detail and molecular basis of inheritance. There we have to study those things. Please see here. Let me explain you this also. Ribonucleic acid. See here the nitrogenous base. Here we have the nitrogenous base which is guanine. Okay, this is connected to, joined by the bond. This is known as N-glycosidic bond with the ribose, and with the ribose on the fifth number carbon we have the phosphate group. And see how the phosphate group is joined to another nucleotides and how it is forming the bond with the 3 number carbon using its phosphodiester bond. Can you see here? So this is the structure of this nucleotide. Okay, you can have the screenshot here, but this is not the part which we will discuss here in detail. We are going to discuss each and everything in detail in the chapter molecular basis. Am I clear? See how they are forming the DNA. They are forming the DNA. This is the single strand of nucleic acid, and again we have the another strand of nucleic acid, and it will form the double stranded nucleic acid. Am I clear? Do you have any doubt till here? So till here we have completed all the polysaccharide and nucleic acid. Okay. Yeah, just waiting for your doubts. If you have any doubts, we can solve now. Chaliye, I think there is no doubt, so I'm just ending the lecture.